Hi, today I am going to present the series of bioinformative talks on different topics. In the first topic, I have selected docking topic. In this topic, I have covered how to develop the approach for protein ligand docking or protein docking and how to use it for different research purpose also. And also it helps to understand how to use the docking softwares or tools, pre-developed docking softwares or tools. So as I mentioned, it covers the protein ligand docking. So we will go for basic introduction, motivation, methods, and conclusions. So it comes in introduction, the basic definitions related to protein ligand docking. The docking could be global and local, rigid, flex and flexible, bound and unbound docking. Then another term is in all kinds of docking, we have the goal to find the binding places and orientation for protein ligands. Then it goes to compatibilities, that is geometrical as well as chemical compatibility. So docking is a basic term in computational chemistry that attempts to find the best matching between protein and ligand. So here a receptor stands for the protein. Local docking somewhat to find the location of the binding site, known binding site. In this case, we only need to orient the ligand in the binding sites or binding region. Then global docking, the general problem includes a search for the location of the binding sites and a search throughout the receptor molecule to figure it out the exact orientation of the ligand and the binding sites on the protein. This could be represented in terms of figure or an sketch. Local docking could be seen here on the left side and global docking could be seen here on the right side. Then rigid and flexible docking. In rigid docking, there is no conformational changes in protein structure, but in flexible docking, there is conformational changes, and then we calculate the binding interactions or interaction energies for when we allow the conformational changes or flexibilities. Bound and unbound docking. In bound docking, the goal is to reproduce a known complex where the starting coordinates of the individual molecules are taken from the crystal of the complex. In unbound docking, which is a significantly more difficult problem, the starting coordinates are taken from the unbound molecules. In terms of geometrical and chemical compatibilities, geometrical compatibilities helps in preventing the overlap between atoms and maximum shape compatibility between the ligand and binding sites. Chemical compatibility, maximum number of chemically favorable interactions and minimum number of unfavorable interactions. The motivation behind the development of a docking tool or approach is to predict the binding geometry of the ligand receptor complexes, which is important for drug design and better understanding of the mechanism of ligand specificity. Docking algorithm needs to be fast and more accurate to check thousands of mutating ligand and binding sites or binding groups. In general, for developing the software or to perform docking, you need to be familiar about the basic databases and softwares. For example, PubChem is the database for small molecules or ligands or drugs. PDB is the protein data bank, so the structures of the proteins could be collected for known proteins from this data bank. In case if it is not known, then collect the primary sequence from uniprot.org and go for structure prediction by using Swiss model or AlphaFold or some other softwares. And also it is important to learn some basic commands And now to map the receptor force fields on a grid box. Energetic evaluation during ligand docking to a receptor molecules could require to calculate receptor ligand interactions for many ligands and ligand placements. Each interaction calculation requires to go through all ligand receptor atom pairs. It is computationally very challenging and demanding. The right side figure is representing the receptor binding site and the ligand placements. In general, there are two interactions which are mandatory to be calculated or included for the calculation of binding affinity. One is van der Waals interactions, another is electrostatic interactions. So next part is mapping the force field on a grid. It is possible to pre-calculate the force field energy of different types of ligand atoms with the receptor on selected points or grid around the receptor. For each possible ligand atom, an interaction grid is stored or interaction energy is stored on each grid point. For any ligand placement at the active site of the receptor, a given ligand atom may be exactly located at a pre-calculated grid point. Then the interaction energy of the ligand atom with the receptor equals the grid point energy. Mapping the force field. Now, if the ligand atom is located somewhere between the grid points, the interaction energy can be calculated by interpolation from the nearest grid points. A simple linear interpolation is enough for this purpose, where one assumes that in the space between the grid points, the interaction energy changes linearly between the values on the neighboring grid points. It is also possible to map forces on the grid. It is then possible to perform energy minimization of the ligand in the force field of the receptor. Now, the advantage and disadvantage of mapping the receptor force field on grid. Any interaction of the ligand atoms with the receptor can be calculated by interpolation from the nearest grid points. This is much faster than calculating the interaction from all the distances of the ligand atom to receptor atoms. Disadvantage. Most common disadvantage is interpolation may not be accurate to add short distance between ligand and receptor atoms. Conformational changes of the receptors are generally not allowed, but in our approach, we have allowed the conformational changes also. So here, how can we account for conformational changes of the receptor? If a receptor undergoes conformational changes, then each time there is a need to recalculate grid energy. Assumption conformational changes can be best described as a collective change controlled by a single variable. Now, how can we account for conformational changes of the receptor stored on each grid point each time the interaction of an atom with the undeformed receptor stored on each grid point? The energy changes upon deformation of the receptor in the collective variable. Docking consists of the movement of ligand and the movement of receptor in the collective variable. Then go for interpolation each time, as it can be seen in the figure. Now, this is the figure or layout to represent for both the cases when you are developing the software for docking. This is left side figure where we collect the coordinates of both the molecules, proteins, and ligand by using a program, and then go for grid setup, then pre-calculation of interactions for each grid point, interpolation, then repetition of the calculation after each conformational changes or deformation, then finally implement it for any kind of test run. But when somebody is using the online docking web server or the softwares, docking softwares by using command lines or some other applications, then it is very simple to collect the protein or ligand and protein-protein. When we are performing protein-protein, Docking, then we collect both the protein structure. If we are performing protein ligand docking, then collect both the structures of protein and ligand, then go for system preparation, perform docking, go for a score, best score, and then collect the docking complexes. Now, we have applied our software developed tool, docking tool, to a kinase protein and a small molecule or ligand. Kinases normally belong to the most drug targets and they are related to cancers and many other human diseases. There are some useful links for protein ligand docking and these are freely available. For example, CA, SEB-Doc2, SwissDoc, then eDoc, Vina Autodoc, etc. Pineball, VMD, Chimera X are the visualization, most common visualization tools. So, in conclusion, we developed our own approach in MATLAB to calculate the van der Waals interaction and electrostatic interactions. In the next step, the program used an algorithm to calculate the same after the receptor undergoes deformation or computational changes. Implementation of interpolation routine and finally calculate the interactions, most optimal interactions. By allowing conformational changes, good solutions could be achieved and thus will lead to novel drug design. Thank you.